This podcast is about reading scientific papers, all about controls. My name is Brad Carter. I'm a graduate student in the Thompson Lab, and this podcast is being put together for our undergraduate journal club sessions coming up in the next week. The goal of this podcast in particular is to learn more of how to interpret scientific figures, how to read the information from the pictures and the graphs and the legends to understand a given paper. We want to go over the basics of interpreting a figure, and I categorize that into two main parts. The first is to determine what the main point of the figure is. What does the author want you to see, to know, to come away with from reading the figure? This will usually involve the title of the figure, which summarizes the overall findings of an entire figure. And you want to compare any information that you can get from the figure to that endpoint, saying, what does this tell me to support this statement? The second major component is to identify and interpret factors that are tested in the figure. And usually, as a gross generalization, these involve manipulations to increase or decrease a specific factor, and usually a series of manipulations for a number of factors to lead to the main point of the figure. And in this case, control experiments are critical. The goal of a control experiment is twofold. It's mainly to isolate the targeted variable. Something happens because of a specific factor. And the controls also rule out additional variables individually. This thing happens because of this factor and not because of X, Y, and Z other factors. So when you're looking at a figure, you always want to start with what's the main point, and then to identify and interpret factors to understand how they make that point and how they conclude from their data the direction that they write about. So let's look at an example and think about this in the context of work in our own lab. So here's a very simple figure where I've diagrammed the experiment on top with some petri dishes and cells and a bar plot on the bottom. And the title of the figure is Figure X, Gene A mRNA is directly regulated by the glucocorticoid receptor. As displayed, the figure suggests that on the y-axis the mRNA level is measured, and on the x-axis shows the condition that's being represented, and would appear to show that there's an increase in the mRNA level when the sample is treated with glucocorticoids or cortisol. However, this figure does not show what its title implies and needs further experiments to make that claim. I'm going to go through some basic points of what else could be considered for the data and how we can add some additional control experiments to address these possibilities and lead us to a more certain conclusion that gene A is regulated by the glucocorticoid receptor. So why doesn't this figure demonstrate the title? The main thing is there's very little control. As displayed, other factors may account for the data besides the hypothesized explanation. That is, the gene may be changed by other factors than the glucocorticoids. So we need additional experiments or controls to isolate the targeted variable, that is, the glucocorticoid regulation of the mRNA, and rule out additional variables individually, so other factors that could change the mRNA levels. When we're thinking about this, a first step is to identify variables. And in this way, reading papers and interpreting them can be somewhat similar to planning and performing experiments in lab. and vice versa. And so by understanding some of the variables that you think about in lab, in your experiments, you can learn and apply them to understanding papers. So what are some other factors that could change the mRNA level? Well, one is the treatment of cells. As displayed in the figure, it just said that untreated controls versus treated cells change the level, but it's known and that just manipulating the cells and putting different chemicals on them and changing the media can sometimes have an effect on gene expression. There also could be other hormone receptors that are regulating the target. For example, even among glucocorticoids, there's two hormone receptors, the glucocorticoid receptor GR, but also the mineralocorticoid receptor MR. And based on the data that's displayed, you can't tell the difference, which one is involved. There's also the possibility of off-target or secondary effects. Glucocorticoids are known to have powerful effects on other RNAs that in turn may be made into proteins and then regulate a second set of RNAs. And nothing in the figure as displayed could distinguish that possibility. And finally, if we're talking about direct regulation, building off the off-target secondary effects point, there's the possibility of RNA versus protein-mediated effects. Is this a direct target that only mRNA is affected, or is it protein-mediated that other proteins are involved or have to be produced to see the effect? So we're going to go through some experiments that could address or test if these factors are contributing to the mRNA change. So let's talk more about controls. The first point is that the treatment of cells or samples of any kind is often compared to what's called a vehicle control and not simply an untreated control. And this is to account for the physical mechanics of the treatment that may alter gene expression. So in this case, we would change the media on the cells and add the solution that the chemical's in, or ethanol that cortisol dissolves in the vehicle, but have no cortisol. So everything is the same besides the cortisol in the treatment. So that's one control. And if the gene is indeed regulated by cortisol, we would expect to plot a similar bar graph as before, but we would plot a vehicle control instead of an untreated control. The next possibility discussed was that CORT activates multiple receptors, the GR and MR. And one way we can address this is to use a selective agonist that targets only one of the receptors. And in this case, we're interested in the GR receptor or think it targets our mRNA. So we're going to use dexamethasone, a synthetic GR agonist. And so we would expect then for a plot, if the gene is targeted by the GR receptor, it would also be regulated by DEX. However, when there's chemicals and drugs used, there's always the possibility of an off-target effect. That is, it's not acting as expected. It's acting through a second messenger or a second molecule. So one way to rule that out is to do the reverse experiment. Instead of targeting the GR for activation, we can target it for inactivation or decrease its activity. One way we could do this is to co-treat or co-administer a receptor-specific antagonist with CORT to show the specificity of the effect is based on the GR receptor activation. In this example, we'll use RU486, the synthetic GR antagonist. So the last control or factor that we identified as variable is whether this effect is directly on the gene for transcription or if it's a secondary effect through another protein. One way to do that is to prevent the synthesis of new proteins and therefore block any effects that are mediated by a second protein in the cascade. So in this case, we would co-administer CORT with the protein synthesis inhibitors, such as anisomycin. Any changes we see, we would attribute to direct targeting by the glucocorticoid receptor. So, and if that wasn't indeed the case, then we would expect something like this, where the co-treatment of CORT with protein synthesis inhibitors does not block the change in mRNA level, suggesting that the gene is directly targeted. To improve upon the initial figure, this bar graph, as shown here, would be much more convincing for the figure titled Gene A mRNA is directly regulated by the glucocorticoid receptor. We have shown in multiple ways that the regulation is dependent upon the GR receptor and is a direct RNA event, not mediated by other protein. So that's the kind of logic that we're looking to develop as we interpret figures. We'd like to be able to go through each piece of data, in this case each bar of the graph, and be able to understand why the experiment was done and what it tells us that strengthens the case of the main point of the figure. So as you're going through your papers, try to think of how each experiment tells you something related to the main point that the author wants you to see in the figure. For some initial practice, created a couple different plots of data that show mRNA regulated by treatment with cortisol or treatment with steroid. And I'd like you to think about the pattern of the data, and we'll discuss at our meeting what we can learn about the gene from this data. So be as detailed as you can. Do this as sort of a homework to get used to analyzing figures. So first we have gene B. What can you tell me about gene B? And we also have gene C. What can you say about gene C in terms of its regulation by CORT? So now we're going to start to apply some of these skills to interpreting an actual paper figure. So I've chosen a figure here to talk about from a Nature article in 2006 relating to microRNAs and neuronal spine sizes. And I'm going to go through each portion of the figure to demonstrate the kind of thinking that we've talked about, and I'd like you to work on developing as you read your papers. So the title of this figure is MIR-134 negatively regulates dendritic spine size in hippocampal neurons. We're interested in seeing how the figure supports that claim. And I like this figure because it has a picture of what they're measuring. This isn't always the case, but it's helpful to visualize any of the experiment and gives us a good starting point. So let's just look at this picture and see what observations we can make. On the left, this figure is using lentiviral vectors in neurons in petri dishes in culture to change the expression of a certain microRNA, 134. So in panel A on the left is labeled GFP. GFP stands for green fluorescent protein. This panel is showing the equivalent of the vehicle control, a virus transfected that has no microRNA manipulation in it, but accounts for the process of transfection and how it affects neurons. The bottom part of the figure is a blown up image of one of the dendrites, and they're wanting you to look at those bulb looking projections and protrusions which are known as spines. That's where the synapses are. In panel B, labeled MIR134, that experiment contains a viral vector that overexpresses MIR134, meaning there's more MIR134 available in the cell. If you look at the blown up portion below, it looks like the spines are somewhat skinnier and smaller compared to the spines in the GFP control condition. Then I'm going to jump over to panel D, where they're displaying what they're calling 2'O-methyl-134. This is a modified anti-MIR, or oligonucleotide sequence, complementary to 134. And what an anti-MIR does is it antagonizes microRNA action. Basically, in this case, would result in decreased amount of MIR134 available in the cell. So it's kind of the opposite of the MIR134 figure, panel B. In that light, it looks like the 2'O-methyl-134 in panel D results in larger spine sizes compared to both the GFP and the MIR134 panels. And the final panel is C, but C is a 2'O-methyl control, or a modified oligonucleotide that does not have the MIR134 sequence. And this is controlling for the process of transfecting in the modified oligonucleotide, showing that the changes seen with the 134 nucleotide are sequence specific. The panel C does not show the increased spine sizes that are seen in panel D, suggesting that changes are from the sequence of MIR134. So we learned all that just from looking at the pictures that they've shown you. If we then look to the graphing, that's exactly what they display. On the left, a plot of average spine volume on the y-axis, normalized to their GFP vehicle control. They quantify that transfecting MIR134 results in a decreased spine volume, and the antagamir 2'O-methyl-134 results in an increased spine volume. And then their 2'O-methyl control on the far right of that bar graph results in no significant difference, as well as the LET7C control. This control is a viral vector containing a microRNA sequence, LET7C, that is not the same sequence as MIR134. And so those two controls demonstrate that the sequence is important for the regulation. In panel F, shown on the right, they're now just demonstrating, is it the width or the length that tends to change in terms of spine size, and they indicate that it appears to be the width that changes based on MIR134 presence, and not the length. The text here is what's known as the legend, and should explain most of the abbreviations and the details of the figure. It should at least allow you to interpret the figure independently of the rest of the paper. The details of the experiments will be noted elsewhere, but most important information to interpret the figure should be included. And so here we have examples of them describing the vectors. They talk about how many spines they counted from how many neurons, and importantly, they detail what their symbols mean. In this case, the asterisks demonstrate significance, It's a very common statistical significance symbol. So if we scale back, here now we look at the figure again as a whole figure. And I hope that it's been helpful to look through each panel to understand what they're contributing to the overall point that Mir-134 negatively regulates dendritic spine size in hippocampal neurons. Or alternatively stated, there's an inverse relationship between Mir-34 levels and dendritic spine size. The more Mir-134 you have, the smaller the spine size, or the less Mir-134, the larger the spine size. So now that I've walked through a figure based on the principles we discussed, I want to give you an individual figure to practice on your own. This is a figure from a 2010 publication in Journal of Neuroscience. It was looking at the effect of cocaine on a specific set of neurons in the nucleus accumbens, which is part of the reward circuitry in the brain and thought to be important in understanding mechanisms of drug abuse. I'd like you to go through this figure. I'd encourage you to make notes on what you find, and we will discuss this at our first session for the Journal Club. I've listed on the right a number of the abbreviations that are in the figure legend to help understand all the details of what's being shown. Just for reference, the panel B displays some electrophysiology measures showing the result of the resulting action potentials from different amounts of current injected into the neuron slices.